Hey guys, welcome back to AP Bio. Last class you learned about the origins of DNA and specifically the people who are responsible for discovering its structure. This class we're going to talk about that basic structure and then also discuss replication of DNA. Before we do that, we're going to cover something called a pedigree. They're pretty self-explanatory but sometimes can show up in the AP exam, so I want to make sure you're familiar with those as well. So I have everything open that you need to be successful for today's class. First thing are our notes. You should open these up and then split screen between this video and the notes so you can fill them in as you follow along. Next is your classwork, which is just a brief worksheet on the different parts of the DNA molecule. And then finally, there's a video you'll need to watch, which will be linked in this post. So make sure that when I tell you to pause the video and watch this, that you do, because this video will give you a pretty solid idea of the different things we're going to discuss and the practice that you'll be doing. So let's go ahead and get into our work. Sorry, presenting it from the wrong slide. All right, so today again, we're going to discuss pedigrees pretty briefly. We'll do some notes on DNA, and then you guys will do a reinforcement sheet to make sure you understood what we discussed in class today. So you guys are going to be able to construct and interpret pedigrees, which show genealogy. That's like traits that are inherited through generations. You'll be able to identify steps in replication and recognize how DNA is related to aging. So to get us started, I wanted to briefly go over pedigrees. Pedigrees show family inheritance patterns, and this goes in with last unit, not the one that we're in right now. So pedigrees are used to identify the genotypes, that's the letter codes that people have, based on the phenotypes in a family. In order to read a pedigree, you just need to know a couple things. The first is that a square like this represents a male, a circle represents a female. A vertical line and a bracket like this indicate that people are the parents of specific children. So these two are the parents of these three here. One mistake I've seen students make that I hope you guys won't is they forget to add this vertical line in the bracket, so then it looks like these people are reproducing with their own children, which is generally not the case when we're creating genealogies. If you have a circle that is not shaded like this or a square, then that person doesn't have the trait that we're looking at. If there's a horizontal line between two people, it represents a marriage or that they had children together. And then if you have a circle or square that is shaded like these two or the one that that's pointing at, that says that the person does have the trait. When we're talking about pedigrees, we can also use them to identify types of inheritance that are displayed. So the first category is autosomal inheritance. This affects both men and women. If it's an autosomal dominant trait, then it usually shows up in every generation. So if you see a pedigree that has men and women, so circles and squares that are displaying the trait, and it's showing up in every single generation, it's probably an autosomal dominant form of inheritance. If you see a pedigree that skips a generation, like this, but affects men and women, then it's probably autosomal recessive. And then finally, if you see a pedigree where the trait is inherited mostly by men or only by men, like if we look at this one here, these guys have it, these women don't, but they can pass it on to their children. So no woman has the trait, they're just carriers for it, these men do. Then that's a pretty good indicator that it is sex-linked inheritance. So that's all you need to know about pedigrees. We'll get into some more practice with them prior to the exam date, but I wanted to put this on your radar and make sure you understood it, since I forgot to add it on to last unit. So now let's get into the crux of today's work. We want to understand the structure of DNA and how it makes copies of itself. This is the first part of your notes, so make sure you're following along as we go through. So DNA was initially established as the molecule of heredity because it had three important factors or traits that led to that. The first is that we know it can store information that relates to the development of the cell. It's also stable, and it can be copied or replicated. And then finally, it has to be able to undergo changes or mutations. Now, initially, this was a topic of contention for scientists because some scientists believe that proteins were the molecule of heredity. That's how we passed on genetic information, based on the fact that there were 20 amino acids. So they thought, oh, we have all of these different traits. There has to be more variability than the 4 bases found in DNA. Now, in 1869, Johannes Friedrich Miescher was the first person to isolate what he called nuclein, which we now call nucleic acids. Those are the things that make up DNA and RNA, or genetic information. Miescher used pus and bloodstained bandages from a hospital to study that nuclein that he discovered, which he isolated from white blood cells. We now know that there are 4 primary bases in DNA and then a 5th that gets added for RNA. Every base has a sugar, a phosphate, and then the base itself, every nucleotide. So our nitrogenous bases are adenine, guanine, cytosine, and thymine, and they fall into 2 categories, either purines, like adenine and guanine here, or pyrimidines, like thymine and cytosine, and you can tell those by their shapes here. Now, there are a couple experiments that actually helped us to prove that DNA was the arbiter of genetic information. It's the place where it was held. And the first was Frederic Griffiths' experiment on transformation. So Griffith was looking at bacterial transformation in rats. And he had 2 different strains of a bacterium. The first was a rough strain, which means it's non-virulent. And we talked about virulence in our epidemiology unit. So you can see he injected a mouse with the rough strain, and the mouse survived. He then injected mice with the smooth strain, which is the virulent strain, and the mouse died. Now, he wanted to see if this information could be transferred. So he killed the smooth strain using heat, injected it in a mouse, and found that it survived. What was interesting about his research was that when he then injected a rough strain, remember that's the non-virulent, with a smooth strain that had been killed using heat, the mice still died. So what that showed is that the S strain bacteria could be transformed into a rough strain. The genetic information in here could be transferred into here and have the same result. Next up, we have Alfred Hershey and Martha Chase. So they did experiments on bacteriophages, and they proved that the molecule of heredity was DNA and not proteins. There are different animations you can watch on this, but for the sake of time, I'm not going to click those. If anyone's interested, pop into office hours or shoot me an email, and I'll send you those to watch. But this does a pretty good job of summarizing their experiment. So they started with bacteriophages, and they either injected radioactive material into the protein coat or into the DNA of the bacteriophage. Remember, bacteriophage is a virus, so it has a capsule, it has the DNA, but it doesn't have much else. Then they allowed those phages to infect bacteria, and then they let new phages and bacterium produce from that result. And you can see over here on the left-hand side where they introduced the sulfur into the protein coat, it was not transmitted to new bacterium or new viruses. None of those were radioactive. But when they replicated that experiment using the DNA with the radioactive material, and they had the phage infect the bacterium, both the bacteria and the new phages that got produced were radioactive. What this means is that the tag on the DNA went into the bacteria, but the tag on the protein did not. So that genetic information that helps with replication is proven to be located then in the DNA. Now, you guys got a pretty in-depth intro to this structure of DNA establishment. You know about Rosalind Franklin, Maurice Wilkins, Chargaff, everyone who was involved in the process. And hopefully you also learned a little bit about these pieces, but we'll go into more detail with them later. Now, from that video, you learned that one of the key things that helped us discover this structure was Rosalind Franklin's radiogram right here, photograph 51, which shows us that double helical structure. Now, you also know that Watson and Crick were credited for this discovery, and if you're interested in discussing this more about the controversy surrounding it, feel free to pop into office hours. But through all of those researchers' work, regardless of who got the credit, we figured out the structure of the DNA, and it is a double helix shape. We call it that because it has two spirals, so two helixes that wrap around one another. Now, if we're looking at DNA, it has a couple of key parts. The bases right here are the steps of that ladder right here. So G, C, A, T form the steps through the center. The sides, or the railing, if we're using this ladder metaphor, are made of sugars and phosphates. And then both of the sides are held together through hydrogen bonds. So the bases bond using hydrogen bonds, which we learned about in Unit 1. Now, when we look at DNA, the interesting thing to note is that the sides are anti-parallel. That means they run opposite directions to one another. The numbers that we're looking at here represent the carbon attached in a ring. So this is the 5 end, this is the 3 end, and you can see that they run opposite one another. This side runs this way from 3 to 5. This one runs 5 to 3. So they're running in opposite directions like that. So again, each side is anti-parallel. One side has the 5 end at the top, 3 at the bottom, and then the opposite for the other with the 3 end at the top and the 5 at the bottom. When we're talking about a nucleotide, remember that's kind of this right here, the part that makes up each section of the DNA. There's the base, those are the steps that we discussed earlier. This one's adenine. The sugar, which is deoxyribose right here, and then a phosphate group attached. So every nucleotide is made of a base, a sugar, and a phosphate. Some other interesting things to know. When we have guanine and cytosine forming bonds right here, they use 3 hydrogen bonds, while adenine and thymine here use 2. And when we're making these complementary pairs, there's rules for how they bond. Guanine always pairs with cytosine, so G to C. Adenine always pairs with thymine, so A to T. The acronym I was taught in general bio was apples are in trees and cars are in garages. So if that helps you remember, that's great. But the other thing about that is that those base pairs are always one purine and one pyrimidine structure. So again, these are our purines, adenine and guanine. The pyrimidines are cytosine, thymine, which are involved in DNA replication, and then uracil, which is in RNA. So adenine always pairs with thymine, guanine always pairs with cytosine. Now we're going to talk about DNA replication, and this is how the DNA makes copies of itself. So replicate is just a fancy word for copying. Remember when we talked about the cell cycle, we know that replication happens during the S stage of interphase, the synthesis stage. So this happens before the cell divides. One important note on replication is that they refer to it as semi-conservative because it is saving half of the strand. So conservative refers to saving, semi means half. So 1/2 of the original strand is saved or conserved, and then we add the pairs to match. Here's an image of what that looks like. So you can see this is our original strand. We cut it in half, and then we build a copy on either side to match those 2 initial strands. So every new DNA strand that gets produced has 1/2 of the original strand and then 1/2 that is new. Here's kind of an overview of what that looks like. So you can see this enzyme right here is splitting the 2 strands. At the fork, we have bases bonding to the new openings. And this runs from the 3 to 5 end. So this part is really important. I want you to pause this video, and then you're going to watch the other video embedded in this post so you can get background information on what we're about to discuss. If you don't pause the video, you're going to be confused. So make sure you pause. I'll give you a second to do that, and we'll come back together. All right, so if you didn't watch this video, again, go back and watch. Otherwise, you're not going to know what's going on. So we're going to talk about all the different things that they discussed, starting with helicase and the replication fork. Remember, DNA helicase, as the video stated, is the molecule that actually is responsible for splitting the original strand into two separate ones. And when it splits, it gets this end here that creates kind of a fork in a road. That's the replication fork. Then DNA polymerase adds nucleotides and binds sugars and phosphates. So polymerase is responsible for building the new molecules, and it travels from the 5 to 3 end. The DNA is called the template strand, so we use that template to build our new one. When we're looking at the building of the strand, there's two separate parts which you learned about in the video. One side is the leading strand. It follows the helicase as it unwinds, and it builds continuously. The other side is our lagging strand. It's moving away from the helicase, and because of that, it can't be built simultaneously, right? It can't be built at one time. It has to be built in different parts. The different parts that are put together are called Okazaki fragments, and they get attached using DNA ligase. So when the lagging strand reaches the when this molecule reaches the replication fork, the helicase is moving in the opposite direction. So it stops and adds another polymerase further down the chain. So you have multiple forks all down the same strand to help with the building of that lagging strand. This is an image of what that looks like, and it has all of the parts, which is why I really like this one. So we have our leading strand here. You can see helicase, remember, is the molecule for splitting the two. So helicase comes along and it splits the strand into two. And when it splits the strand, then we have two different strands that form at the replication fork, which is this part right here. So you have your leading strand and your lagging strand. Remember, the leading strand is running from 5 to 3 end, and it just follows the helicase along. So it attaches all of these free nucleotides at one time. Then you have your lagging strand. Remember, the lagging strand can't be built at one time because it's moving from that 5 to 3 end. So because of that, we have these Okazaki fragments that form. Those fragments attach to the different parts that are built, and then ligase comes along and kind of clips them on to attach them to the different parts. Remember, we'll have more replication forks further down the line, but we're always running from 5 to 3. So the other parts we need to be familiar with, DNA primase, remember, is the one that builds the primer. So it tells the process where to start, and it adds a little RNA primer to that section. Once the primer has been added, then DNA polymerase runs along that line and adds more free nucleotides, as we see here. DNA polymerase running along this line here as well. Ligase is responsible for attaching those parts, those Okazaki fragments that we have. And then once that's done, we have two separate but identical DNA strands. The last thing you need to know is that sometimes this process doesn't work the way it's supposed to. So DNA can undergo changes called mutations. And usually we have different repair molecules in our body that can catch those changes and correct them before replication is completed. Sometimes they don't catch them. So when that doesn't happen, we have 4 major types of mutations. The first is called a substitution or a missense. That's when we switch one base or letter, and it creates a new protein as a result. The second is an insertion or deletion, also called a frame shift. This is when you add a whole new letter in or take one out. Insertion and deletion are, generally speaking, more dangerous than substitution because they're changing the whole sequence of protein after, not just single base. Then we have duplication. This can amplify the amount of gene that's produced. And then copy number variation, where it just repeats specific sequences of that strand. Go ahead and pause here if you didn't get those down because you need those in your notes. All right, now for your notes, this is the part that I'll be grading aside from just for completion. So I want you to take however long it takes, probably 5 minutes, and go ahead and answer for each of these 10 questions what the different parts are. So if we've got an enzyme that unwinds DNA, you know that we talked about 3 to 4 different enzymes. Remember, the one that's splitting the strand is the one that's unwinding it. So helicase is the one that you would put here. Go ahead and answer the other 9, and I'll be checking those in your notes. And then when you finish with that, all you need to do is complete that reinforcement worksheet. So you'll upload your notes with those 10 questions answered and the notes filled out. And then your reinforcement worksheet will act as your exit ticket or your kind of assessment score. So this is classwork, this is assessment. Both grades will help improve your score in class if needed. As always, thank you guys for watching. If you need any help, you can stop in on office hours and I'd be glad to help you out. That's 2:45 to 3:45 p.m. You can also shoot me an email and I'll try to answer you as soon as possible.